The overall goal of this procedure is to demonstrate the formation of bacterial streamers in a microfluidic device with micropillars. This is accomplished by first fabricating the microfluidic chip. The second step of the procedure is to culture the tested bacteria, in this case Pseudomonas fluorescens. The final steps are to assemble the experimental setup, inject the bacteria into the microfluidic chip, and collect data. Ultimately, results can show time evolution of streamers through fluorescence microscopy images.
Visual demonstration of this method is critical, as the different steps are difficult to learn because of the interdisciplinary nature of the experiment.
This procedure requires silicon wafers with deep reactive ion etching. The photoresist on the wafers should be washed off. Begin with silanizing the master mold. Add 2 or 3 drops of trichloromethylsilane to a vial and place it in a desiccator along with the mold, design upward beside it. In 2 or 3 hours, the mold will be silanized. During the silanization, prepare the PDMS. Mix Silgard 184 silicone base with a curing agent at a 10:1 ratio. Then degas the PDMS under a vacuum for about 2 hours. Now transfer a silanized wafer to a holder and pour the PDMS over it, ensuring that no bubbles are formed. Allow the PDMS to cure on the wafer at 80 degrees Celsius for 2 hours. This fabricates a PDMS stamp from the silanized silicon mold. When the curing is completed, peel off the PDMS stamp with the aid of a cutter. Then cut the stamp into a micro